Hello students, welcome to LEELA'S tutorial. Today let us discuss about pharmaceutical importance of milk proteins. Normal human milk consists of protein casein, right? Apart from that, it consists of various nutritional qualities which are required for the infant or the newborn baby, right? Which cannot be replaced by any other products. But here, by using animal biotechnology, they have produced an transgenic animal in such a way that its milk is equivalent to the mother's or a human mother milk, right? Let us see how they have produced this transgenic animal. So first, they have selected the gene, right? They have selected the gene which codes for alpha-lactalbumin, right? The gene will get expressed in order to produce this alpha-lactalbumin. So this is the desired gene of interest which has to be introduced into the cow, right? So how they are going to introduce that gene? So here they are selecting the superior quality female, right? Superior quality female cow, and they are collecting the oocyte or the ovum from that cow. Then after collecting the ovum, they are allowing that ovum or the oocyte to undergo maturation in tissue culture media. Once the oocyte or the ovum is matured, here the gene of interest which codes for alpha-lactalbumin is directly injected into the pronucleus of the ovum. So now this ovum is allowed to undergo development. Then the developing embryo is introduced into the foster mother, right? The mother, here the foster mother bears that baby or carries that baby for the complete gestation, right? After gestation period, it will give birth to the progeny. So now once the progeny is produced, now this progeny consists of the desired gene of interest. So now this progeny, what happens? Now this is having that gene which codes for alpha-lactalbumin. But we have made the gene in such a way that it is active only in the mammary glands. Though the gene is present in the progeny, it will undergo gene expression only in the mammary glands. Mammary glands are nothing but milk producing glands. So the gene expression will occur only in the mammary glands, so that the milk produced by these glands consists of this alpha-lactalbumin, right? So now the milk consists of this specific protein alpha-lactalbumin. It will be analyzed, fractionated, then it is purified and made it into a powder, then packed and marketed, which is having same nutritional, or you can say complete balanced nutritional quality, same as the human mother milk, right? And this cow which was producing this milk, transgenic cow which was producing this completely nutritionally balanced milk, is named as Rosie, right? The cow was named as Rosie, whose milk is same as the human mother milk, which consists of all the balanced nutrients. So in this way, they are able to produce the transgenic animal so that it can have some required or the specific proteins in its milk. By simply consuming the milk, it acts as that remedy, or you can say it acts as a medicine. So here topic itself, pharmaceutical importance of milk protein. That means how the protein present in the milk is used in order to decrease the disease, right? So in the same way, we have another animals, the transgenic animals, which are having ability to cure certain diseases by using their milk, right? So first one is sheep and the pig. So here the transgenic sheep and pig were able to produce tissue plasminogen activator in their milk. What is the use of tissue plasminogen activator? Right? It is an thrombolytic agent. Right? The tissue plasminogen activator is a thrombolytic agent where it breaks down the blood clots, right? And allows free circulation of blood in some parts of the brain. Right? So here, if the sheep, transgenic sheep and the pig are producing this tissue plasminogen activator, when it is consumed, it results in avoid of thrombosis disease, right? It will not allow any blood clots to get happened in the brain. Okay, next coming to the another transgenic animal, cow and the sheep. Here the transgenic animals, they were able to produce fibrinogen in their milk. Again, what is the use of fibrinogen? So here fibrinogen, which is naturally produced in the liver, it will circulate in the blood. Wherever the injury has occurred in that particular region, the fibrinogen will get converted into fibrin by means of thrombin, and then the fibrin results in formation of a network-like structure or the clot, so that the bleeding can be stopped. So here this fibrinogen, which is obtained from the milk of cow and sheep, are used in wound healing process. Next coming to the another transgenic cow, which was producing collagen and collagen 1 and collagen 2. Again, what is the use of collagen? Collagen, it consists of certain proteins, the collagen helps in healing the joints. Okay, and also it helps in skin elasticity, right? So, and also in the stiffness, right? That means here the wrinkles, everything can be cleared. So here by drinking this transgenic animals milk, the diseases such as tissue repair, rheumatoid arthritis, right? This is related to the bone. Tissue repair related to the skin. All these can be cured by using this transgenic milk. Next coming to the goat. Here the goat is able to produce Pro542 protein. So here it is an Pro542 protein is an tetravalent CD4 immunoglobulin fusion protein, right? So this protein is having an ability to inactivate the primary HIV, right? Human immunovirus. The primary human immunovirus can be inactivated by using this protein. So here this goat's milk is used in case of HIV or the AIDS patients. Next coming to the another transgenic animal, cow, where it is producing lactoferrin. Lactoferrin is having multiple functions, right? Some of the functions like the low iron content in case of pregnant women, they can use this lactoferrin milk, right? Otherwise, the infants, even the blood related infections can be avoided. So mainly here this lactoferrin enriched milk from the transgenic cow is used in gastrointestinal tract infections, right? In order to cure these infections. Next one is another transgenic animal. It is rabbit, where it is producing alpha glucosidase. So here alpha glucosidase is used in case of Pompe diseased patients. So in case of Pompe disease, there is accumulation of the starch or the sugars, right? So what happens? The sugars which are accumulated or glycosides, right? They will be broken down by this enzyme, right? So when it is broken down by this enzyme, again it will help the small intestine to get absorbed, right? So in that way the Pompe disease can be cured by having this alpha glucosidase containing milk from the transgenic rabbit. Next animal is transgenic mouse, which is able to produce malarial antigen MSP1, right? So here MSP1 is merozoite surface protein. So mainly it is used in curing the malaria. So malarial parasite is Plasmodium. One of its stage is the merozoite stage, right? So here at that stage the animal can be killed as a vaccine. It works as like a vaccine where the malarial antigen which is produced in the milk is specific to the merozoite surface protein 1, right? So it will go and bind to this protein 1 of the merozoite surface, right? As a result, it will kill that parasite at that particular merozoite stage, and also malarial fever can be reduced. Is that clear? So in this way we have so many which are transgenically improved in order to cure the diseases, right? And also their products are having high importance in the pharma industry also, right? So apart from these transgenic animals, if you have gone through any other animal which is having any beneficial or the pharmaceutical effect, you can just post it in comments or in WhatsApp or Facebook.